Our next speaker is Jonathan Gootenberg, who is a graduate student at Harvard and jointly in Feng Zhang's lab and Aviv Regev. I do have to admit a conflict of interest here. I work very closely with Jonathan, and you might call us work spouses, and I hope your girlfriend is okay with that. So he'll be talking about our work in characterizing Castor DNA or C2C2 and our more recent applications in creating a detection platform with it and mammalian transcriptome engineering toolbox.
Thanks, Omar, for the great introduction. And I know I'm the last thing between everybody and lunch, so I'll try to keep it brief. But today I'll be talking about some things we've been doing in the lab, in vitro and in vivo, using what you've been hearing about today and yesterday a lot, Castor DNA, which is shown here, or crystal structure from Liu et al. So really doesn't need an introduction, but Cas9 is just a marvelous tool that's really allowed us to do so much. Of course, it can do all these amazing things using its DNA editing capability. By cutting DNA, we can edit the genome, or we can even record things and really do amazing kind of manipulations. But beyond that, just as a DNA binding platform, it can obviously bind to the genome and upregulate or downregulate transcription. We can even turn it so we can modify the epigenome. And going beyond perturbation, we can actually fuse it to GFP and in live cells see how the genome changes in structure. So as this kind of as a framework for what we can do with a nucleic acid binding protein, it kind of makes you think, what can we do with RNA targeting tools? So today I'll be focusing on Cas13a, which was previously called C2C2, but credits to Eugene for the unification of the nomenclature. But of course, there's also Cas13b, as David talked about. But they're kind of a newly discovered class of RNA-guided RNases. And they're somewhat similar conceptually at a first pass to Cas9. So you have, this is your generalized type 6 array, type 6A array, I should say, with Cas13a, where Cas13a is expressed, and the CRISPR array is also expressed. It's processed, as Alex showed, by Cas13a itself into mature cRNAs, which are loaded into Cas13a, then used to target invading phage RNA, either perhaps from RNA phages or perhaps from RNA from DNA phages, leading to cleavage. So you can think of it conceptually as quite similar to Cas9 to a first extent. But of course, there are caveats. So when Omar and I were originally characterizing Cas13a, we found that it had a very interesting characteristic, which we termed the Collateral effect. And it's a triggered promiscuous cleavage. So to walk you through it, without the targeted ssRNA, if you just have other RNA in the solution, which was fluorescently labeled in the paper, but of course it doesn't have to be, and you incubate it with Cas13a, you don't get cleavage, which is great because we'd like our CRISPR enzymes to have specificity and to not be cutting things up willy-nilly. But if you actually add in the targeted RNA, and then you have this fluorescently labeled collateral RNA, then you get, as you would expect, cleavage of the targeted RNA. But you also get cleavage of these other RNAs in the solution. So this was actually very interesting. And because we had it fluorescently tagged, we could actually use this as a signal that we had this RNA specific here. So it's a very interesting property. And you can see that we and, as Alex talked about, other groups have turned this into an RNA detection platform. So here you can see species of RNA with our kind of chosen blue sequence there. And by incubating with a Cas13a loaded with a cRNA targeting that species, we can actually activate collateral cleavage. And the way we can detect that is we can use this cleavage reporter, which has a fluorophore on one end and a quencher on the other. And upon cleavage, you can actually separate those two and have the fluorescent activity. So it's a great way to detect RNA. It is fairly sensitive, as you'll see later. You can go down to the femtomolar range. And it also leverages the specificity of these CRISPR enzymes inherent. But can we actually push it farther? So there's been a lot of acronyms today, and I'll credit Aaron with the fact that we all have to design acronyms. But we wanted to actually combine it with an extra step to create a platform that had specificity and sensitivity together. So we called it Sherlock for specific high sensitivity enzymatic, that's the Cas13a, reporter unlocking. And what we did is we took our collateral-based detection and we coupled it with an isothermal amplification process called RPA. And you can probably guess that RT-RPA stands for reverse transcription RPA. And what RPA is it's conceptually quite similar to PCR, except it occurs only at 37 degrees because you can have enzymes do the work that heat would do in PCR. So what we can then do is take either DNA or RNA and expose them to this isothermal amplification to create a large amount of DNA. Then we can actually turn that back into RNA using RNA transcription. And then once we have our RNA, we can detect it very specifically with Castor DNA and leading to both a very specific and sensitive tool. So I've said a lot of things about the sensitivity. Well, how sensitive is it? So if you just look at Cas13a, you can get down to the kind of femtomolar range. And that's 10 to the negative 15th, or about a quadrillionth. But if you actually add in Sherlock, you can get down to the automolar range. And that's 10 to the negative 18th. And we, just to kind of like calculate it, 1 microliter of a 2 attomolar solution contains about a single molecule. So you can see that we drop off like a little bit below an attomolar. It's because there's nothing else there that we can really detect. So this isothermal amplification really pushes it down into this range. And we can do that for RNA, but we can also do it for DNA. So here you can see that we essentially have constant detection. Here we were actually quite lucky, and we kind of stochastically sampled a single molecule. But once we get down below that, then we just can't see anything. So when you're doing these things, there's a bit of randomness involved because the basically quantum nature or singular molecule nature of these samples. But you can see that we're really getting down to actual single molecules. So we're really excited by this platform. And there's a lot of applications that we actually wanted to pursue. I don't have time to discuss them all today. But I'll focus on a couple that I think highlight both the sensitivity and the specificity, as well as other great aspects of the Sherlock platform. So first, we wanted to kind of see if we could detect an emerging pathogen. So Zika has been a kind of nice test for the sensitivity of these assays. And in collaboration with Jim Collins' lab, who previously used CRISPR in a system to detect Zika, but from another system, we wanted to see if we could actually detect lentivirus containing either Zika or the related virus dengue. So we exposed it to Sherlock, where we did RPA and then Castor DNA detection. And you can see that for the dengue virus, which is kind of about 60 to 70% similar, we don't get any detection at these large amounts. But by particle counting, you can actually see that we are getting down significantly to single viral particles. So in this kind of mock Zika assay, we can detect single molecules of the Zika genome. So another great thing about the Broad is that there's many clinical groups here. So in collaboration with Pardis Sabeti's lab, we could actually acquire human serum or urine that actually contained Zika. And they actually reverse transcribed it and RNase H'd it for us because they usually use these for qPCR reactions. And then we could do RPA and put it into Sherlock. And you can see that we can detect essentially 1 copy per microliter when we're putting in 1 microliter into the Sherlock reaction. So this is also a nice proof of principle. One thing that we're pushing forward is that this purification step is required when you're doing qPCR because of PCR inhibitory kind of species in the biosample. But we're excited to kind of push this forward. And we have some preliminary data showing that we can directly sample urine and detect Zika in under an hour. So it's a very nice platform for when you have things that might be inhibitory for PCR. To push it a little farther, we actually wanted to say, well, we have enzymes. Can we make them into something portable? So Jim Collins' group has a great history of freeze-drying enzymes. And so we wanted to see if we could freeze-dry the Cas13a onto paper. And this would create something that was stable and that could be hypothetically used as a point-of-care diagnostic. It's also nice to note that RPA itself is already freeze-dried. So here, all the components could be cold chain independent. So we used the same mock Zika test before, and we saw that we could go down to about 10 or to 20 attomolar. So freeze drying gives us a slight hit to our sensitivity, but we're still sensitive enough to detect 10 copies per microliter, and this is in a format that could be shipped across the world and then reconstituted at the point. So it's kind of an exciting prospect. We also thought, well, these Castor DNA enzymes have been shown to be fairly sensitive. Can we take some applications that highlight really the sensitivity of them and the specificity to really create a sensitive and specific platform? So we wanted to actually detect cell-free DNA. So mutant cell-free DNA is something that occurs quite often in cancers. When you have a cancer and it's shedding DNA, this is actually mock DNA because we couldn't get clinical samples in time for the paper, but it is fragments, and they're from BRAF V600E, and they have a single point mutation. And by designing guides to either target the wild type or the mutant version of BRAF, we could maybe assay the cell-free DNA, even when it's at very small proportions and large background of wild type. So we wanted to sense with our wild type sensing guide first. And you can see that for various levels of wild type DNA, here we go down to about 95%, it's essentially uniform. And you can't really see the BRAF there because you're 95 versus 100. It's not a very stringent test. But if we use our actual V600E sensing guide, you can see that for as low as 1 in 1,000th contamination of the wild type with a mutant, that we can actually sense significantly an increase in signal that's indicative of a potentially cancerous mutation being present. So it's a great test of both the sensitivity and the specificity because we're detecting very small quantities with single base distinction. And we're excited to move this forward. You can imagine many other applications that combine these two qualities beyond just cancer. So we think it's a very nice platform for detecting and sensing all these kind of nucleic acids that surround us. So there's been a lot of talk about in vivo today, of course. So we'd also like to take Castor DNA and put it into mammalian cells. And the obvious application, at first at least, is to do it for transcript knockdown. So we actually adapted Castor DNA. We stabilized it with a superfolded GFP. And then we put our CRRNA or guide under a mammalian promoter. And we wanted to see, could we target a reporter vector? And we actually chose a dual luciferase reporter where we could target one luciferase and then measure both. And by the ratio of the two luciferases, we could have an indication of targeting. So we actually wanted to see if we could use either nuclear or cytoplasmic Castor DNA because you could imagine context when you want to target both. And you can see that both the cytoplasmic as well as the nuclear, which actually performs better, Castor DNA could target and cleave the luciferase reporter about as well as RNAI. So to us, this was a very good first indication that we could actually get RNA targeting in live mammalian cells. So we wanted to kind of leverage the reprogrammability, so we actually did a tiling screen. So what we did is we could take a transcript and computationally design tiled guides across it. And we hadn't, at the time, had a lentivirus construct, so we actually did arrayed plasmid cloning. And then you can clone these and co-transfect them into cells with Castor DNA, where you can read out expression by qPCR or luciferase assay. So we targeted a couple genes. One is cyclophilin B or PPIB, and you can see that there are regions of very good targeting in certain areas, and we think that these may be indicative of secondary structure. So it's kind of a simultaneous readout of both that we can target these genes, but also that there's structure to them. We also wanted to target a nuclear gene because RNAI is not very good at nuclear targeting because it's really a cytoplasmic mechanism. So we targeted MALAT1, which is a nuclear non-coding RNA. And you can see that similar to PPIB, we actually get targeting down to 50% knockdown. And we're pushing to optimize this even further. We also wanted to actually bring it to a completely different system. So in collaboration with Dan Voitas, we actually wanted to target plants. So here you can see that we actually took rice protoplasts and we generated protoplasts and then could transform and read out expression. And you can see that for 3 genes targeted, including EPSPS is the target of Roundup, for example. These are metabolic genes involved in plant growth. We can get knockdown around 70%, and it works for several different guides. So we see it as a nice platform for targeting across different organisms. As Alex mentioned, Castor DNA does cleave its own array. So this raises the possibility that we can actually express a single array, like you might with CPF1, and use it to target multiple genes simultaneously. So we designed an array with 5 different spacers. And you can see that we can actually target and knock down all 5 genes at comparable or better levels of the pooled gene. So here's all the individual guides pooled together. And we can actually get targeting that is equivalent just by the native processing abilities of Castor DNA. So I've talked a lot about targeting with Castor DNA. You might be familiar with RNAI, which is another RNA knockdown tool. So we wanted to compare to RNAI. And so many of you may have heard that RNAI is fairly nonspecific, but here is proof of that. So we did transcriptome-wide sequencing on shRNAs, either non-targeting or targeting our luciferase gene. And in red, you can see that there's this luciferase knockdown, which you can see represented by its distance off the central axis. But you can also see there's all this fuzz. And these are genes that are being slightly upregulated or downregulated, which can lead to a lot of off-target effects. And that's one issue with RNAi-based screens, for example, which is that they have off-target effects, which makes them not as highly powered as, for example, a CRISPR screen. So if you compare this to Castor DNA, you can see that Castor DNA is much more tight, that you don't have these off-axis elements, and that we actually basically get a single line. And cloudiness versus not cloudiness is not really a method of rigor, so we can quantify differentially expressed genes. And what you can see is for RNAi, targeting 3 different genes, you can get up to anywhere from 500 to over 1,000 off targets. But for Castor DNA, we could not detect any. So you can get essentially equivalent levels of knockdown without the off-target effects of shRNA. Lastly, we've talked a lot about knockdown, but can we use Castor DNA for other things? So you can imagine using a catalytically inactive Castor DNA or decastor DNA for many different applications. One we were interested in was the imaging of stress granules. And actually, in collaboration with Alice Ting's lab, we designed a negative feedback Castor DNA that contains a zinc finger and a crab, wherein it's unbound, and in the nucleus, it represses its own transcription. But when it binds to the transcript, it will actually translocate to the cytoplasm. And we should be able to see a big increase in cytoplasmic signal, and therefore the ability to separate signal from noise. So if we just look at the Castor DNA in 2 targeting conditions with either beta-actin, or another beta-actin guide compared to non-targeting, here you can see that in the 2 targeting conditions, we get much more expression compared to non-targeting, and it's cytoplasmic compared to nuclear, which is a good indication. And applying this to stress granule tracking, so we can stain for G3BP1, which is a marker of stress granules. This is by immunofluorescence. You can see that we get substantial overlap in these beta-actin targeting guides, both here and here, but no overlap when we target with a non-targeting RNA. So we can actually target these transcripts, which are known to localize to stress granules, and see a stress granule response. So in summary, we can do both single molecule detection that is specific and portable, but we can also do in vivo RNA targeting, and it is as well specific, but it's a versatile RNA binding platform. So to bring it back to kind of what you can do with Cas9, we're starting to think about what we can do with Cas13s. And so we actually had this slide yesterday, as you might remember, but we can enhance translation and repress translation. We can modulate splicing. We can use fluorophores or NLSs to assess or alter localization. And then we can also, of course, target and degrade RNAs. So we're excited about creating a versatile platform using the technology, and we're really looking forward to what we and others in the community can do with it. So with that, I'd like to thank the many people who made this possible. Of course, Zhang and my co-advisor, Aviv, the many people in the lab, of course, Omar, and our collaborators, Jim Collins, who we did much of the Sherlock work with, Eugene Koonin, nothing else is needed, and then Pardis and Deb, who helped with Sherlock applications, and Alice and Dan, who have been helpful with the mammalian and plant targeting. So with that, I'd like to open up to any questions people might have.